So, my name is Alyssa Price, and today we're going to be exploring core protein's role in plastic biodegradation and waxworms for my final Genetics 312 project. So, the plastic problem. Every year, 19 to 23 million tons of plastic waste leaks into our world's aquatic systems. Plastic can take 20 to 500 years to decompose. It is not naturally biodegradable, therefore the decomposing process is largely based off the environment. The Great Pacific Garbage Patch is an example of the crisis we are in today. The patch of floating plastic is larger than most of the countries in the world. As shown on the left, it is about a third, a little bit more, of the size of the United States. So what are waxworms? Waxworms are the larvae of the waxworm moth, also known as the Galeria melanella. In the wild, they live as pests in bee colonies. In bee colonies, they eat the bee cocoons, beeswax, as well as their honeycomb. They have four main stages of their life cycle. Waxworms will remain at their larval stage for a period of time depending on the temperatures they are exposed to. In warmer temperatures, they tend to undergo metamorphosis quicker. Waxworms' biodegrading properties. So waxworm saliva is capable of oxidizing depolymerizing polyethylene, or PE. PE is one of the most produced and common plastics used in packaging. Waxworms' capability to degrade PE is really important due to the biodegradation of plastic because PE is one of the most resistant polymers due to its dense structure. So some research has been conducted to show that waxworms are capable of oxidizing and depolymerizing PE is waxworms were fed PE instead of their honeycomb diet for a couple of days. As the larvae devoured the plastic sheet, their excretions changed, indicating the presence of different chemical wastes. Various tests have also been performed to extract the bacterial DNA for sequencing and species identification. They found that the wax moth larva also plays a role in PE degradation in their microbiome. The four salivary enzymes that have been identified in waxworms are Demetra, Sibelius, Ceres, and Cora. All four of these biodegradable enzymes belong to the hexamerin family. We also know that these enzymes biodegrade plastic because of their ability to oxidize PE. The ability to oxidize PE is important, again, because it's the most produced in common packaging plastic material. So our research questions. First up, what information is missing about the function of the saliva enzymes? We are missing the specific code of the salivary enzymes, specifically in waxworms. We are also missing the enzyme diversity in a variety of waxworms in the wild versus commercial waxworms. So why are we specifically focusing on the core protein? Well, the core protein is also the most abundant protein in waxworm saliva. Therefore, we are likely to get more data from it. We also do not know how similar or different our genetic sequences will be. Nobody knows this information because the existing sequence for the core gene that is published is the sequence from only one individual worm. And what part of the core protein are we focusing on and why? We're focusing on exons 3 and 5 of the core protein. These are likely the most conserved parts. The similarities and differences show us what the essential genes are for life in the waxworms, and what are the specific traits to each one. We want to compare the similarities and differences between these exons in commercial and wild waxworms from a variety of different areas in North Carolina. We also wanted to see if their different climates and food sources made a difference in the protein's genetic makeup. So, obtaining our waxworms. We obtained the wild waxworms from beekeepers whose hives have been infected by them. They graciously donated us these hives, and more specifically the waxworms that had infected their hives. The commercial waxworms were from a pet store near NC State. So thank you, beekeepers. So extracting our DNA from the worms. First, we weighed our waxworms and gathered our initial data to put into our group drive. On the top left is the wild waxworm, and on the bottom left is our commercial waxworm, and mine specifically. We dissected their heads and added a buffer to them, and ground them up into it in a tube. The reason for this is because we are wanting to sequence exons 3 and 5 from the core protein in their saliva. We then centrifuge the liquid from the heads multiple times to isolate the more concentrated DNA of the head and get rid of the unneeded material. If we have enough DNA from our extractions, we can now run PCR. PCR amplification. To prepare our PCR, we need to dilute our DNA concentration and add different reagents for each exon we are looking at. These master mixes will reduce any chance of error caused by cross-contamination. Then we run the PCR to amplify the large number of copies in our specific exons that we are coding for. We then can observe our PCR amplification by using gel electrophoresis. So PCR amplification analysis is via gel electrophoresis. If we see bands in our gel, it means we have enough DNA in our PCR to analyze these exons using the Sanger sequence. Initially, my PCR had too low of a DNA concentration to be seen by gel electrophoresis for all of my samples, so I had to go back and double the DNA extracted for the PCR. On the right is showing my initial PCR amplification. As you can see, there are no bands for the exons of my specific waxworms. And on the right is the rerun of my PCR amplification, and you can see the bands outlined in it. And the second time I ran my gel electrophoresis, I was able to see all the bands for both exon 3 and 5 on both the commercial and the wild waxworms. So once this is done, we know we can continue our analysis and we can run a Sanger sequence to get the genetic code of the exons. So Sanger sequencing. First, we need to desalt and clean up our PCR products. This is necessary to remove leftover enzymes, primers, buffers, etc. so it doesn't get mixed up in the sequencing. And we use Sanger sequencing to read the small targeted regions of the Cora protein, which are exons 3 and 5. And after running our Sanger sequencing, I found out I was unsuccessful in the sequencing of exon 3 on the wild worm in the reverse direction and exon 5 on the wild waxworm in the forward direction. This was because the sequence was too busy and I could not identify the proper genetic code from it. The sequences that did turn out good were then compiled in our class data. And on the left is an example of a good Sanger sequence that is not too busy and you're able to identify the proper genetic code from it. And we first make sure our sequences match up with our waxworm genome using BLAST, and then we run MAFT. So our sequence alignments. These are our sequence alignments. It's a section of it, of the middle section. Exon 5 on the left is our area of high homology, and then we have the exons 3's area of high homology on the right. So what does our sequence alignment tell us? After compiling our class data on the Cora protein, we were able to see a vast array of commonalities and differences. At the beginning and ends of our sequences as a group, there were a lot of differences. This can be due to the length of the sequence, frame shift, point mutations, etc. But the middle parts of the exons had high homology, which is what was in the last slide. When observing the differences between the wild and commercial sequences, I found that the wild waxworm sequences varied the most. Although most of the time the sequences were similar, the wild waxworms overall had a higher rate of variation in their genomes. I believe that this is due to the variations in the waxworms' locations. The different locations tended to have a greater difference in their sequences as compared to the waxworms from the same location. The commercial waxworms generally had the same sequence throughout, but this can be due to the environment they were in and their diets as well. Future directions. If we had more time to work on this research project, I would continue sequencing a variety of commercial and waxworm core proteins on exons 3 and 5, but I would begin collecting a larger sample size of data. I would first begin collecting wild and commercial waxworms from different areas of the United States, but if there's enough resources, I would try and collect wild and commercial waxworms from different parts of the world, especially since the world is a variety of different climates. If I had more time to work on this project, I would also further analyze the conserved proteins or enzymes in the waxworm's saliva, especially the 4 core proteins in their saliva. I believe that the more we can observe the genetic data for multiple conserved enzymes in the waxworm, the more likely we are to find their biodegradable plastic properties. I would use a variety of waxworms from different locations if I was to do this research as well. Thank you for listening to the research we conducted, and I hope you enjoyed it.